I think you'll see I come at it a very different angle from our first two speakers, and completely different end of the brain too. So I hope you enjoy. There's a little bit for everybody, I think. So I want to start with just thinking about this remarkable ability that we have to adjust our actions based in goals and plans. And I would define this particular thing that we can do as, oh boy, I can learn this, I swear, cognitive control. And this particular thing could come in handy if you're doing something like planning out the meal that you want to make. You have to plan a whole series of actions, let's say, to go to the store and then finally click and finally create your meal. And so I want to define sequences as just a series of items that you're doing in a specific order. So it can be actions, it can be motor actions, it can be thoughts. And these things are pretty ubiquitous in our lives. You have to do things in an ordered way. It wouldn't do you any good to start drying off the dishes before you've actually put soap on them. And what I hope to show you a little bit about today is that monkeys also have these kinds of sequences. So how do we start to think about how the brain controls these kinds of sequences? So just to illustrate the fact that this is a unique capability, if you have patients who have frontal lobe dysfunction of some kind, often they can do pretty complicated tasks where you can ask them to, say, for example, categorize by shape or color on these cards in an individual trial. But when you start to put together all of these things that they have to keep track of to, say, for example, cook a meal on their own, they're incapable of doing that. They're incapable of independent living as a consequence. And in fact, this has been called the most important and also the most neglected problem of cerebral physiology since the 1950s, is understanding this idea of serial order and how we put things together. And right now, at least, this is also something that AI and robots also aren't very good at. They fail kind of epically in these ways. Who knows, maybe they'll catch up eventually, but for now anyway, this is something that's pretty unique to us. Okay, so to be a little bit more concrete about the kinds of sequences I'm going to be talking about, I want to use this as an example, and that is making coffee, something that probably many of us did today, or tea if you prefer that. This you can consider as your goal. You can break it down into a series of sub-goals, and you can break that down into a series of abstract tasks and break it down further into motor sequences. Now, the reason why I call these tasks abstract is because they're not dependent upon the exact stimuli or the exact series of motor actions that you make. If somebody puts the coffee beans in a different place or the milk is on a different shelf in the refrigerator, you can still thankfully make your coffee just fine, including if somebody completely changes the coffee maker on you. This happens to us all the time when we travel. So motor sequences, on the other hand, the way you might think about those as an example is playing the piano, playing the scales on a piano. If somebody took and rearranged the keys on a piano, you would be completely lost. You would not be able to complete that. So the majority of the day today, I'm going to be talking about these more abstract level sequences. So how do we think about the neural systems that are underlying it? Well, in a cartoon version of the human and the monkey brain, you might start to think about looking for where's controlling these kinds of sequences in areas that are already associated with flexible control. And that's perhaps in contrast to where you might classically think about looking for motor sequences in places like the motor or premotor cortex or basal ganglia. So just a brief overview of how we study these sequences in the lab. As I already mentioned, we work on monkeys, and they of course have lovely complicated behaviors that are sequential, but then they have the huge advantage of being able to do the kinds of electrophysiological recordings that we heard about earlier today. And then we also do work in humans. They also have great behaviors. We work in healthy and in clinical patients. And we do techniques like fMRI and TMS, non-invasive way of stimulating the brain. And a major focus of the lab is to then actively bridge across the two species. And the way we do this is by doing fMRI in non-human primates. And so we're really hoping that we can work towards going beyond the idea of behavioral analogy and really thinking about the functional homology across the species. So just quick overview of what I'll tell you about today. I'm going to tell you about how we as humans control task sequences, some of the computations that are underlying this. I think some of the dynamics that I'll talk about here will tie in with some of the dynamics we've already been talking about today. A little bit about obsessive compulsive disorder and what could be going on there. And then I'll transition to talking about the monkeys in MRI and what we've been most recently working on was in the electrophysiology. Okay, so just a quick refresher for thinking about this abstract task sequence level. So how do we do this in the lab? Our task is pretty simple. On every trial, you're going to see an image, and you have to categorize that image by pressing a button. And what you're judging is the color or shape of that image. Pretty simple. So how do you know which judgment to make? Is by, in the beginning of a block of trials, I would show you an instruction on the screen like this: color, shape, shape, color. So then when the first image comes up, you would have to remember, oh, I'm supposed to remember color, so I'm going to press the 1 button because it's red. And the second trial, I'm going to see a blue square. I have to remember I'm making a shape judgment, so I'm going to press the 2 button because it's a square. And so on and so forth, you would go through the sequence. I just want to note that I'm showing you the size of the stimuli changing. That's irrelevant to the task. It's just to give us some more variety in the number of stimuli that we have. Yes?
Instruction is always there.
So this is one screen, people see it, and then it goes away. And so they have to remember where they're at in the sequence. Thank you for asking. And so the way this looks across a block of trials, and this is relevant to the question you just asked actually, is that in a block of trials, people have to repeat the sequence, and they don't have external cues about where they are in the sequence. So what this creates is this internal sequence boundary where they have to restart the sequence. And these sequences come in 2 forms. So if you generalize color and shape judgments to A's and B's, this first form is ABBA, something like color, shape, shape, color. And we call this a complex sequence because it has 2 task switches in it when you switch from color to shape or shape to color. The other kind that we do is a simple sequence. It only has 1 switch in the middle of it. Importantly, though, when you string them together, the first position in the simple sequence is actually also a switch. So the total number of switches and repeats are balanced across the sum total of what people are doing. Okay. So how do we know people are actually doing what we ask them to do? They're performing these things as sequences. There's some really nice behavioral markers that we have of that. So what I'm showing you here are reaction times collapsed across the 4 positions in the sequence. And what you can see is a few hallmarks of the things that they are doing. I'll break it down for you. So the first is that you see these elevated reaction times at the task switches. So for example, in the complex, you have color, shape, shape, color. Whenever you're switching, you have an elevated reaction time. And that's in contrast to what you see at task repeats, which is a lower reaction time. And probably what you all saw right away is this increased reaction time at position 1, which we call sequence initiation. And the reason why this is a great marker for us is that if you think about the complex sequence, so color, shape, shape, color, this color, when they do that judgment, for example, is actually a task repeat. So if you were just going to see a reaction time based on the fact that they were repeating, you would expect a reaction time down here, but that's not what you see. So this initiation is really unique to the fact that you have to initiate the whole sequence. So where are we looking in the brain? So we're interested in this part of the brain here, the lateral, along the middle frontal gyrus, the lateral cortex. And why we're interested in that part of the brain is because when people are doing static decision-making tasks where they need to incorporate a hierarchy of information, this area of the brain is the area that's representing that highest level of the information. So that's the area that we were interested in, because now essentially we're giving people that hierarchy of information in sequential form. It's not static. Because based upon where they are in the sequence, that's how they know which decision they have to make. So this is the area here. If you take a slice through cortex here, I'm looking at the rostrolateral prefrontal cortex. And what do you see in the task when people are doing it in this area is that you see a ramp up in this is bold activation as people are going through the 4 steps of the sequence. And it does reset to some extent. And then we can talk more about that, but it does reset at the beginning of each sequence. If I look in the whole brain as to where is showing this kind of ramping dynamic, it's not the whole brain. There is a network of areas. It tends to be in this kind of frontal parietal control network kind of areas, but it's pretty unique. And I am going to focus on the rostrolateral prefrontal cortex today, but that's not to the exclusion of these other areas existing. Okay, so there's a lot of ramping there. Is it unique to the idea that you're involved in a sequence? Do you have to have this dynamic to be reacting to this task? So this is another area that we wanted to take a look at. This area right here, it's premotor dorsal. It's also known as Broca's area. So what we wanted to see there is, do we see differences that are associated with these sequences differently? And it turns out, yes, that we do. This area just shows basically a shift in activity. It doesn't show ramping. So it just dissociates between the simple and the complex sequences. So we have different kinds of sequential responses in different areas of frontal cortex. It's not like the whole frontal cortex has this particular dynamic. And you'll see here in the whole brain that that response between complex and simple is relatively isolated to this pre-PMD area. Okay, so big question is, what is the ramping for? Well, first question is it necessary? If so, then you might expect that it would be potentially carrying out the sequence itself. If it was doing that, then it would be necessary throughout the entire sequence. Or is there some sort of transient process that's engaged such that you only need it at specific points along the sequence? And one idea that we had is that maybe you had to check at particular points where there was an increase in uncertainty. So what do I mean by that? Well, if you define a sequence as just a series of states, and I just mean steps here, I know state can be a loaded word sometimes, then you might imagine that each time you transition from one step to the next, you might accumulate a little bit of uncertainty in such a way as the fact that by the time you get to the end, you have more uncertainty about your position in the sequence than in the beginning. So this was one of our initial ideas going into these experiments in terms of what this dynamic was doing. The way we approach this is to use transcranial magnetic stimulation. It already came up, but just very briefly, it's non-invasive. It's an electromagnetic field that's being very briefly induced over the scalp of the cortex and over the cortex, and it briefly disrupts neural activity in a relatively small space. Think on the order of under 10 millimeters or so, and over a relatively short amount of time. So within 10 milliseconds or so for the single pulses that we'll be using. So when we stimulated so this is the exact same task that I already showed you. We stimulated at a point in time that was between when the stimulus turned on and when people made their response, just a single pulse. And the way we set this up in the task is that we only stimulated at most once every sequence. So most once every 4 trials. So sometimes position 2, sometimes position 4, and so on. I'm showing breaks here between the sequences just to illustrate. People, again, had no external cues as to where they were in the sequence. Okay, so what did we see? So first, we were stimulating RLPFC and this other sequence-involved area, but with a different dynamic, pre-PMD. And we did 10 different stimulus onset asynchronies because we actually didn't know when would have the greatest effect in terms of what we were looking for. And here's what we see. So this is irrespective of position in the sequence now, across the 10 different stimulus onset asynchronies, what we see here is an increase in error rate at earlier time points for the rostrolateral prefrontal cortex, and an increase in error rates at later time points for premotor dorsal. So already a little hint that maybe there's a hierarchy between these 2 areas. But now what about the task? So I'm going to show you the difference in error rate between stimulated and non-stimulated trials. And I'm going to collapse across that middle position. So I'm just showing you 3 points here, because what I really care about is the dynamic from beginning to end. And what you see is there's this ramp up in error rates between stimulated and non-stimulated trials for the rostrolateral prefrontal cortex, and almost the opposite dynamic for the premotor area. Now, we went into this experiment not necessarily expecting this. So we actually did the whole thing again. And this time we added on another control region, the rostromedial prefrontal cortex. And the reason for that was that when you deliver TMS stimulation, it feels like somebody is flicking you in the head. And we wanted to control for the fact that the sensation is going to be very similar between here and here, but it's going to potentially be different here. So we wanted to make sure we had as good a possible active control for the fact that what we were actually looking at was the result of stimulation and not just the physical sensation of receiving TMS itself. So we see this again, we see this ramp up in error rate as in RLPFC, and it's different from, and the result in rostromedial prefrontal cortex is not significantly different from 0. Okay, so what have we learned? This area ramps up during sequence performance. It's necessary for these sequences. And it seems like it suggests that there's a transient check during sequential tasks to stay on track. It is part of a broader network, but it makes the intriguing suggestion that you're going to be more vulnerable to making errors on something that you're keeping track of. So this provides a potential explanation for what might be happening to those who have frontal lobe dysfunction, and that they're going to potentially lose track of where they are. Okay. So in this next part, I'll talk a little bit more about a series of experiments very quickly to try and get better at what's the computation that's actually going on with that ramping dynamic in the frontal cortex. And just to refresh you, this is our example. I'm going to pick out some pieces of it as we go along. And I will say I'm going to fly through these a bit. I'm not going to try and unpack all the experiments. I'm more than happy to talk about any of the details if anybody is curious or wants to know. Okay, so first experiment was to look at position uncertainty and unpack that a little bit more. So what if you don't know, did you put the sugar in your coffee? Do you need to have multi-level decision making? Do you need to have this hierarchical structure to the decision or not? Do you need to be able to see the position cues? Sometimes you can tell where you are by looking at what you're working with. Sometimes you can't. And what about if you change the goal or the reward that you're going to get at the end? How might that influence these dynamics? Is that a part what's going on there? Okay. So to address this first question about uncertainty, what we did is we gave people a task where we gave them clues about where they were in the sequence with the idea that that would reduce the uncertainty. And oh boy, I assure you these people's brains looked very healthy when I put them on the slide. So I'll just tell you that the rostrolateral prefrontal cortex, again, we observe ramping activation here. So that's good. But our key question in terms of do they change if you have clues or no clues, the answer was actually no. So in this kind, there's lots of ways that you can manipulate uncertainty. But in this case, it didn't seem like whether or not people had clues was actually changing that dynamic very much. Okay, so now what about multi-level decision making? Well, what if I take it away? What if I just ask people to keep track of a series of objects? Phone, flag, beaker, piano. Remember that order. Tell me if it's in the right order as I show them to you. That's it. Pretty simple. We again replicate the fact that we see ramping in RLPFC with the addition of a lot more visual cortical areas, probably because you can now observe the sequence directly. It's not a series of abstract decisions. And then So we really were able to say, okay, you don't need multi-level decision-making in order to observe this, even though it's in this very rostral part of the brain. Now, what if you don't have position cues? Do you still see it then? So we basically gave people the same task, asked them to remember a series of objects, but then we pretended like there was something occluding those objects. And people had to remember what those objects were supposed to be. And then occasionally we'd show them one and say, is this in the right order? We see ramping again, good. But we don't see a significant difference between the occluded and the visible. So we take this to say that even without position cues, this is still there. So this dynamic seems very robust. Our last question really is a little bit different from those in the sense that we really wanted to think about, well, what happens when you change the reward? There are ramping dynamics that have been observed in the brain that have to do with anticipation of reward, for example. Could it be some form of that we're looking at? And so what we did is we gave people the same task of remembering an order of objects, and we just changed it so that there were high reward sequences and low reward sequences, so we could see the difference. We replicate ramping. And what we see here, let me just take you through this. This is bold signal change again through two sequences. High reward in red, low reward in blue. You see in the first sequence that I'm showing you, the ramping is pretty much the same between these two. But when you get to the second sequence, which is now closer to the end of the block and closer to when the reward is actually going to be delivered, you see this dissociation where you see the high reward continue to ramp up and the low reward actually look like it takes a little bit of a downturn. So it is influenced by reward, but only towards the end. It doesn't look like that this is the only thing that's driving it. And probably it's modulated by it, but only when it's most relevant. So it's not an explanation for the dynamic in total. Okay, so that was really fast. Just to recap, what I showed you is that we have this ramping dynamic. It's there regardless of uncertainty. It's present without multi-level decision-making, without position cues, and it's modulated by the goal and reward. So then, yes.
So you argue that there's no effect of certainty or visual cue. I don't know. I was looking at the error bar. They looked like they would be seen. They were not.
They were not.
Okay.
Yes. We wanted them to be.
But they were kind of moving in that direction.
Yeah. So the thing about the one with uncertainty is it actually, even if there was a significant difference, it would be a difference in the opposite direction that we would have predicted. So we would have predicted that there would be less ramping with less uncertainty, and we were seeing maybe a trend in actually the opposite direction. So we were pretty wrong. So what we really think this dynamic is doing is tracking or monitoring state information through time. And there's some really nice modeling work that we didn't do showing that ramping as a dynamic can be in a really efficient ordinal code precisely because you can ascribe a unique level of activity to each position in whatever it is that you're tracking. So this is where we're at right now in terms of the way we're thinking about this. So maybe I'll pause and ask if there are any more questions before I'm just going to show you a little bit about OCD before I start talking about the monkeys, but I'm going to move on from this segment. Yes.
The ramping is averaged over all of the repetitions of the forward.
It is, yes.
Every time you get back to the first element, it's low again.
Yeah, so I alluded to this, but when we look at the dynamic across the entire block, it looks like there's potentially a larger ramp that's going on across the block, and then this is kind of a sawtooth riding on top of it. So it's not a pure reset at each position. Okay. So how do we think about this when it's potentially going awry in something like obsessive compulsive disorder? Well, the reason why we started thinking about OCD is that you can observe obsessions and compulsions where people are repeating a whole series of actions. So for example, somebody might constantly check that the door is locked before leaving their house, or check the stove while they're in the middle of cooking for whether it's on or off. And that would have the effect of actually preventing them from reaching the goal that they want to reach. So we thought if we were looking at a signal that has to do with tracking or monitoring kind of high-level sequential information, then perhaps there could be something disrupted in the system when thinking about this population of people. Okay, that's not the only reason. So the circuitry could overlap. This is a cartoon illustration of some of the areas thought to be involved in OCD, and it overlaps with a lot of the areas that I've been telling you about. There's been theoretical work that's postulated that there's these structured event complexes, and when you start talking about structured event complexes, they basically boil down to being sequences. So we thought, okay, we might be on the right track here. You have deficits in non-sequential cognitive control tasks. So all of these, and you see symptom severity correlations with these kinds of deficits. So all these things put together, we came up with a couple of alternative hypotheses to test. So the first thing, which probably will not be surprising, is that we thought that abstract sequential control deficits, we would see those in the form of reduced activity in RLPFC and reduced ramping activation there. Alternatively, there are areas that have been very strongly associated with cognitive control, mostly via task switching tasks in the dorsolateral prefrontal cortex and anterior cingulate cortex, and we could potentially see deficits there. Okay? And we thought that we would see symptom severity correlations with those deficits. So we're using the exact same paradigm that I already showed you. And now we have two groups of people, those with OCD and healthy controls. And so again, I'm just showing you the exact same reaction time over the 4 positions in the sequence. And just to remind you, we have sequence initiation, and then I'm also going to show you switch cost, which is just the difference between the reaction time or error rate on the switch trials and the repeat trials. So I showed you here the reaction times for healthy controls. It's going to be a lot of lines, but I'm going to overlay those for OCD. And what you'll see is that they're basically the same. They follow the same general pattern. There's maybe a little bit of a shift where folks with OCD are a little bit slower, but for the most part, it's the same. The picture changes a little bit when we look at reaction times. Error rates. What we see here is there is a deficit such that the switch cost is much smaller in OCD than in healthy controls because they don't have this drop in error rates on the repeat trials the same way. And that change actually does correlate with symptom severity. So there is an element here. And so how this relates to our hypotheses, well, actually, it doesn't necessarily point to sequential deficits, but more towards this idea that they have a deficit in task switching. Okay, so the main question really was, are we going to see deficits in RLPFC? Just to remind you where it is. So in this region of interest, we looked at onset activity, and we saw that there's no difference between healthy controls and OCD with just onset activity, but there is a slight correlation with symptom severity. So it doesn't completely rule out the fact that this area might be involved. Ramping activity, which we think is this, controller of, sequential ordering here, turns out there's no difference and there's no correlation with symptom severity. So we're left with a bit of a question mark here, given that it seems like there's a hint that maybe some of the activity they could correlate with symptom severity, but not much difference in the ramping that we've been talking about. And maybe suggests that instead what we're looking at is something that's primarily loading on this more general cognitive control and task switching circuit. So to look at that a little more closely, what I'm showing you here is the total network of areas, and I'm sorry, this is another graphic that didn't make it, that is responding with ramping dynamics in OCD in red and in healthy controls in green, and the overlaps in yellow. But what you'll notice is that there actually isn't that much yellow. There isn't a lot of overlap. So even though when we're looking in our region of interest, there isn't much of a difference, it really appears like there's just a different set of areas that folks with OCD are using to complete this task. And when we directly contrast it, what we see is two areas. This area in the superior frontal sulcus, which is right on the posterior border of dorsolateral prefrontal cortex, and the rostral anterior cingulate cortex. So basically, what this means to us is that probably what we're looking at in OCD, even though we can manifest as these sequential deficits, it really looks like it's more potentially driven by the circuitry that's involved in more general cognitive control and in task switching. So this just generally sums that up, which is basically what I just said. And there's one big caveat, though, is that in this task, people are switching tasks in sequence. So we can't completely rule out that there isn't something to the fact that people are engaged in this more complex task that means that these deficits are showing up in this way. And so we really think that we need to potentially expand the idea of these neurobiological models for sequences. And we have some other work that I didn't have a chance to go through today, where these abstract sequences can be useful diagnostically. For example, we show that anxiety disorder dissociates from OCD and healthy controls in this same task that I just showed you. Okay. So last couple pieces here. Now I'm transitioning to talking about the monkeys and how they fit into this picture. So, I've spent all this time, I've hopefully convinced you that rostrolateral prefrontal cortex is doing something like monitoring state information through time, and that we can see sequence-related disruptions in clinical disorders. And what we really want to do with monkey fMRI is eventually get to a place where they're performing the exact same task as humans, so that we can go forward and test functional homology and look at these dynamics at the neural level. So where do we start with this? The first is just a basic question of, do monkeys have anything like an equivalent of the kinds of activity that we're seeing in this really far rostral area in humans? And a place to start is the dorsolateral prefrontal cortex in monkeys. Why? There's been lots of electrophysiology studies that point in as this being a good direction. You see serial position tuning in dorsolateral prefrontal cortex in motor sequential tasks and in series of stimuli. And you see non-sequential abstract rule representation there. So there's a good possibility that these things overlap in this area. And when you look at the anatomy, it seems like, well, maybe, we're looking at an area up here and thinking about dorsolateral prefrontal cortex here. But when you're thinking just strictly about anatomy, that leaves out the function, right? This is beautiful work that has now been followed up on by folks who are thinking about the functional connectivity. So asking the question of where correlates with other areas in a similar manner. And so this area, RLPFC, is in this maroon area here. And it actually turns out that where the functional connectivity is most similar is an area that's deep in the principal sulcus in area 46 in the monkey. And that is actually connected the most similarly to human rostrolateral prefrontal cortex. So that's where we're going to start. And we're going to start with two basic questions. Does monkey area 46 represent abstract visual information in sequences? And are there sequence-related ramping dynamics there? And we're going to try and answer this with the simplest sequence tracking monitoring task. I say simplest in quotes because anybody who works with monkeys knows that monkeys are never simple. And of course, there's lots more to unpack here as we go forward. Good. So probably, even if you can't name that tune, or even if you were subjected to my horrible singing, which I thought that you might be for a moment, if that hadn't have played, you might recognize that. And why do you recognize it, no matter what? Is because it has this really characteristic pattern. Same, different, same, different. This is an example of the kind of task that we're doing with the monkeys, is we're having them look at sequential rules. So this is a rule-based sequence now. So it's not dependent upon what exactly the stimuli are, because you could still identify that no matter what. So how we do this with the monkeys, they're going to be scanning. So they're in their little monkey chair. This is a drawing that my graduate student at the time made of one of our monkeys actually sitting in the monkey chair. They're in the Sphinx position. So they go into the bore of the magnet and they look at a screen at the end of the bore of the magnet. And what we have them doing is fixating a central spot. There's no report in this task. And what they're looking at are fractals. And those fractals come in a particular pattern, which is same, different, same, different. And we can use any fractals for that. So we have a couple of different stimulus pools. We have the habituation set, which you're seeing illustrated here. And then we have another set that we use for deviance from what we've habituated to. And then the sequence types are pretty simple in that we have a particular rule that they're habituated to, and then we pull from this deviant pool to do new items with the same rule, rule deviance, number deviance, or double deviance. And in a whole block of trials, I know there's a lot of colors and information here, but the basic idea is that each block starts with habituation, and then you have infrequent deviance in subsequent blocks. There's only one kind of deviant per block, and it's only 6 out of the total 30 trials. So what we're going to do, because this is a no report task, is use deviant responses in the brain to index the fact that there is a sequence representation there, with the idea that if you break the sequence, you change the rule, then whatever area of the brain was tracking that information is going to show some sort of response. So the way we do that is with these kinds of comparisons, where what we're looking at is the response to rule deviant compared to new items of the same rule. And the reason why that comparison is important is because it controls for the fact that these are both infrequent sets of images. So it's not just an oddball response. Whatever response we see here is going to be to the rule and not to the fact that it's new images, because both sets of the comparison have new images. We do the same for number deviance. So, oh boy. You can't see it very well, but the area that we're looking at here is in the fundus of the principal sulcus. We're looking in area 46. So there's a circle there and a circle there on the left and on the right. And so this is what we see in that area. We see significant responses to this rule and number deviant. Maybe a bit more on the right than the left, but there's no statistical difference between the right and the left. So it's a little hard to make any strong statement there. And if I just show you, I'm not just cherry picking. If I show you what these contrasts look like in the whole brain, what you see is that you see DLPFC for the rule deviant and the number deviant, and actually the only place in the entire brain that shows responses for both kinds of deviance is this area in the dorsolateral prefrontal cortex. So now the other question is, well, do we see ramping there? And we use similar model as to what we used in the humans, with the same idea that if we see a change in the ramping, then that will indicate to us that there's a change in tracking that information. And so same sets of contrasts that we were looking at before, same ROIs. And what we see is, yes, we do see a significant response here, where we see an increase in activity in this ROI for the rule deviant compared to the new item, same rule, less so for number in this case. And if you look in the whole brain, I know it's hard to see, I'm sorry, graphics all got moshed. The dorsolateral prefrontal cortex is there. And as you might have thought from looking at the bar graphs, not so much in number deviance. Okay, So we had two main questions. Does monkey area 46 represent abstract visual sequences? We think we can say yes. Are there sequence-related ramping dynamics there? Yes to that as well. So we're setting up a situation where we can start to look at and think about the actual homology across the species. This is lots more work to be done. I know I've just put these things kind of side by side for now, but we think this is a pretty good start. There's potentially some interesting things to think about as well in terms of thinking about what's going on across the hemispheres. There's a little bit of a left bias for ramping in the monkeys. The same thing is also true in humans, so there could be something more to think about there as well. All right, so next question we had here is, well, how specific functionally and anatomically are these responses in area 46? Sequences are composed of many things. Even these relatively simple ones have time, image, level of familiarity. There's lots of different components. Area 46 is also not a unitary construct. It's actually rather large. And so there's some recent really beautiful work that parcellated Area 46 into 8 different subregions based on the cytoarchitectonics, the functional connectivity. And so what we wanted to start out doing is we wanted to look at the Area 46 that we showed, well, that others showed was most similar and functionally connected. Our previous ROI here down in the fundus, that's this green area here, posterior 46F for fundus. And we wanted to compare that to a directly adjacent area, P46V, because that area shows very different connectivity to the area that we were looking at. It's a little bit more ventral, but it has many more visual connections, actually, than this P46F area. So we wanted to know, is it all of 46, or can we really segment within this area? So the way we did this is by looking at a combination of sequential and non-sequential tasks. So the sequential task I already showed you. We compared this directly to a non-sequential task that kept all the same structure, but just shuffled the order of the images. And again, we're comparing these two areas here. And here's what we see. So these green lines are our friend, P46F. And this data is not new. I'm just showing you the rule and the number deviant responses that we saw before. What's new is these blue lines, this more visual region. What you see is it doesn't really care when there's a deviant at this particular, at this level, at the sequence level. And that's in contrast to in the no sequence task, where now you don't see changes in P46F, that fundus sequence region, but you do see responses in this more visual, more ventral region. Same thing, so when you change the timing or when you change the image. So we can dissociate between those two areas. Another feature that we wanted to take a look at was familiarity. Lots of beautiful work in the visual cortex showing lots of changes due to familiarity. Not as much is known in the frontal cortex. So again, we're going to look at these non-sequential tasks now to look at this. And I showed you the grouped version, and we had images with high exposure. And probably you all already noticed that there's a set of images that you haven't seen before, low exposure images. And we did the same thing now and also just jumbled up the timing as well. So we have a grouped and ungrouped version of the same thing. And again, in our same two regions, P46F and P46V, we see a very similar story where P46V is really the one that's showing responses to the change in exposure, and P46F, this more sequence region, is not. Okay, so in this last little bit, I'll tell you what we're doing in the electrophysiology, which is the newest piece, because we had to get through all of this before we got here. Precisely because we are doing functionally targeted neuronal recordings. So what I'm showing you here is inflated monkey brain. This is anterior, posterior, and then right and left. And there's these little blue splotches on it. That is this monkey's contrast of the rule deviance. And there's these little red dots that are showing you our recording locations with respect to that. And you might be able to see it a little bit better here in this slice through. We're using a chamber, we're using V probes. And we went in with a few very simple hypotheses to start. That we will see neural firing rates that'll dissociate between the sequence types, that we'll see sequence-related neural ramping, and that we can anatomically localize these deviant responses to areas that are similar based on the bold activation. Okay, so in terms of what we see, so this is the breakdown of the kinds of responses that we see. And this is about 650 units across 2 monkeys. So we have responses to deviant type, we have responses between the habituation and deviant types, and then we have about 20% of our responses that are to ordinal position. And just to show you what these look like, these are just some example neurons. The responses are pretty varied. So it's not like there's a unitary kind of response here. I want to emphasize that these are examples. So this is just time 0 here is image onset. This is a cell that responds more in this phasic way to new items of the same rule over rule deviant. We also see the opposite, which would be greater response to rule deviant over new items of the same rule. And then this is an example of what a deviant preferring cell over habituation looks like here. Again, all aligned on one of the image onsets. And then this is an example of a cell that responds primarily to ordinal position. So we have all these different kinds of responses. So proof of concept, and this is very much a work in progress at this point, that we at least see firing rates that dissociate amongst the sequence types, and that we see ordinal position responses, including some ramping activation. It's not at all the majority of what we see, though. So, you know, this would be something that's very interesting that I think touches on some of the other things that we've been talking about today already, that I don't think that the ramping that we see in bold is at all going to be individual neurons that are showing that dynamic, even though if you squint, so you can see, for example, in this cell, position 1 is the highest, but then position 2, 3, 4 does show a ramp up in activity. That's not the majority of what we see. So there has to be something else that's leading to that in bold. And that we're just at the beginning to try and figure out. So then the other piece of this is where we see those responses. Does it at all line up? There's been beautiful work in the visual cortex, various visual cortices that has shown that that's true there. This is the front of the brain. This is no man's land. We don't know, right? So what I'm showing you here, looks a little noisier, this contrast, because this is now a single animal. And what I'm showing you here is our recording locations in yellow, this rule deviant over new item, same rule in warm colors, and new item, same rule over rule deviant in cool colors. I'm just going to blow up that section in the square. And what you can see, hopefully, is that where you see red colored dots localizes pretty nicely to where you see this blob of activation here. And outside of that, we actually don't see significant responses very often. Right? So first pass, it looks like we can at least say that these responses are co-localizing to the same areas that we're seeing. So this is one monkey, and then I'll show you our other monkey. So same idea. These are recording locations for him. And if I zoom in on it, then you see that primarily these deviant responses are occurring where we see the differences in bold and not outside of those regions. Okay. So very quick cartoon recap. I talked to you about how we saw responses in the brain in humans of the rostrolateral prefrontal cortex that we really feel like are tracking this kind of high level sequence activity. And that may be disrupted in things like OCD. There could be different set of areas engaged. We think we have an inroad to thinking about how this is functionally homologous to an area in the dorsolateral prefrontal cortex in monkeys, area 46. And we've started looking at our recordings from there that at least shows a first pass correspondence to the bold. And some other things that I didn't have time to tell you about today is we can also think about inferences in the other direction as well, where we can take what we're seeing in neural recordings and the kinds of responses we're seeing there, and then go back through this chain of inference in the other direction and look for responses in the fMRI as well. So with that, I just want to thank the lab, my funding, and thank you all.
Yes, so thank you very much. It was fascinating talk, and I am interested also about this, like when you mentioned this obsessive compulsive disorder, where you were thinking, did you also consider specifically people with Asperger syndrome?
We didn't, no.